Thank you all very much for joining the Pediatric Dermatology Research Alliance webinar on gene therapy for large chronic RDEB wounds. I'm your host, Jen Dawson. I'm Pedra's Outreach Manager. Before I begin, I want to say thank you very much to Abeona Therapeutics, who is sponsoring this evening's webinar. Okay, a couple of housekeeping items. For optimal viewing, if you're able to watch on a laptop or desktop, that would be the best. We are recording this webinar, so if you find yourself having technical issues or have to jet out, you can find a recording of this on Pedra's website. And we will be having a live Q&A session at the end of Dr. Tang's presentation. So please use the Q&A button at the bottom of your Zoom screen. We will be answering those questions live. And that brings me to introducing your speaker this evening. It's my pleasure to introduce you to Dr. Jean Tang. She's a professor of dermatology at Stanford University Medical Center. She's the principal investigator for the EB-101 pivotal phase 3 VIITAL study. She has spoken at Pedra meetings in the past as well as Pedra webinars, so it's an absolute pleasure to have her back with us this evening. Thank you so much, Dr. Tang.
Hi, everybody, and I will share my screen if that's okay with you, Jen?
Yep.
Okay, so hopefully you guys can see this okay. It's great to be here and great to talk to you about the phase 3 study. I can see a few of the participants so far. So hi, Yoni, hi, Dawn, hi, Sharon. These guys know more about EB than I do, so glad that you're able to participate. So as an outline, I'm hoping to go over these slides for the first 20 minutes and really use the rest of the time for questions and answers. So among Pedra, I don't need to tell you guys too much about recessive dystrophic EB. These patients, unfortunately, have a genetic defect in collagen 7 that leads to an absence or loss of function of anchoring fibrils, and that leads to fragile, blistered skin that eventually leads to open wounds that obviously cause a lot of pain and suffering. As you guys know, there are lots of different subtypes, especially the generalized severe, where, patients have horrific wounds that cover a large surface area of their skin, as shown in this picture. And many of our patients, because of the chronic inflammation and wounding, do pass due to metastatic squamous cell infections and even anemia and cardiac abnormalities. The economic burden is huge. There's no FDA-approved treatment, and oftentimes in EB clinic, some of our patients stop coming because over time the physicians really don't have anything new to offer, and they are the experts in their own wound dressing. And all the Mepilex, Mepitil, all the fancy dressings we use surprisingly cost upwards of $245,000 a year, because there's so much wounding. And all of us know who take care of EB patients, we do all these prior auths and filling out, especially in California, Medi-Cal forms just to get insurance to cover these things. The humanistic burden obviously is huge. There's individual variations, but I'm very privileged to work in this patient population because they've certainly taught me a lot of strength and resilience. And even going through COVID, I feel like, one of my patients said, I've been training for sheltering in during COVID because that's what I do all the time. So, as hard as this year has been, I just can't imagine what it is like to be an EB patient or a caregiver. So, with wound care, all the wrappings and dressings, they really don't address the underlying cause of disease. I certainly know in the literature in EB clinic, we use allograft products, derm graft products, and those work okay, especially for smaller wounds. But really for large chronic wounds, those are the wounds that are most difficult to heal. So the ideal RDEB treatment would really target the genetic cause of the disease and really restore collagen 7 function by correcting keratinocytes. We want to focus on large chronic wounds because in interviewing and talking to patients, these wounds are large, and they're much larger than recurrent wounds. And I know this nomenclature is a little bit new. We talk about chronic open wounds as wounds that are large and have been present for at least 3 to 6 months, depending on what study you read. Whereas recurrent wounds, we define as wounds that might heal and then open up, heal and then re-blister again. And so in some of our natural history studies, we've certainly seen that pain and itch and quality of life is severely impacted really by these large chronic open wounds. So what I'd like to tell you about today is some of the results from our published phase 1/2A study. This was really an investigator-initiated trial started at Stanford. We enrolled 7 participants, and all of them were treated from 2013 to 2017. So the longest patient has been followed for 6 years post-treatment, and the shortest has been 4 years. That served as the scientific basis for the phase 3 VIITAL study, which is taking these EB101 grafts. And this is one of the first registration studies for autologous skin grafts in this disease. You will see the clinicaltrials.gov notation here, so you can look that up for more details. The goal of the phase 3 VIITAL is to enroll 10 to 15 participants, and each participant hopefully will have an average of at least 2 or 3 grafted wounds. So at the end of the study, 35 chronic wounds can be treated and assessed. A little bit more about EB101. So in this diagram, here's a shadow picture of a person. And so we take 2 biopsies, and they're standard punch biopsies, 8 millimeters in size. We ship them overnight to the manufacturing facilities in Abeona in Cleveland, where they isolate keratinocytes, expand them, and use a retrovirus to introduce the full-length collagen 7A1 gene. The keratinocytes take 10 to 12 days to mature, and the whole process ranges from 25 to about 30 days. The patient then either is allowed to stay in the Stanford area or flies back in about 26 to 30 days, where the patient undergoes general anesthesia, the chronic wounds are debrided, and the new autologous EB101 grafts are now sewed on. So this diagram basically shows you that over time, what we see is healed wounds in EB101 treated skin, and in serial biopsies, we do see linear basement membrane zone staining of collagen 7 and also anchoring fibrils. The key thing that a lot of folks ask is, you showed pictures of patients with such huge wounds. So far for the phase 3, you got to remember that the CMC, or the chemistry and manufacturing process, is very much a stringent protocol. So for this study, 2 biopsies lead to the manufacture of 6 of these autologous grafts, and they're about the size of an old iPhone. That's the case for the phase 3 protocol. In the future, if this is approved, then physicians can potentially do more biopsies and generate more grafts, or you can do serial grafting. So, there is this potential to certainly treat more areas of skin. I'm going to show you a few photos in patients treated in the phase 1/2 study. So on the left-hand side, if you can see my cursor, this is a large chronic wound on a young woman's arm, and C and D are the wound areas that are grafted, and C and graft D are shown here, both of them EB101 treated wounds. And on her antecubital fossa, we did not graft this area, and this was our untreated wound control. So at 9 months, you can see the majority of the EB101 graft is intact at C and D, whereas our control, the untreated wound, remains open. On the right-hand side, what we did was take a close-up photo of a baseline wound, and the dots here are the Canfield camera technology that basically shows the area of the EB101 graft. You can see nice wound healing at 3 and 6 months later. The lower panel shown here is a direct immunofluorescence staining. At baseline, there's no green line, and that green line basically shows you an antibody staining to the NC2 portion of collagen 7. So at 3 months, at a serial biopsy of the grafted area, you see nice collagen 7 exactly in the basement membrane zone. This collagen 7 has been detected in serial biopsies at 6 months and later as well. So we also got more aggressive. We started treating larger areas of wounding, and this is a woman's back, and she complained of 10 out of 10 pain, 10 out of 10 itch, and this caused considerable stress on her upper back. And you can see multiple wounds that never heal, and perhaps the opened erosions migrate in different areas. But in this area of her upper back, there is always wounding. So in this area, we basically quilted 3 grafts labeled A, C, and D. And you can see a little bit of erosions around the edges of the graft. And over time, we still see persistent wound healing all the way up to 24 months, also with collagen 7. So as a summary of the data, we're presenting the percent of wounds with 50% wound healing, and this is the gray column, or 75% wound healing, and that's the blue column. And this is the number of months after treatment. So we're focusing at 6 months because this is the study endpoint. The FDA asked us to look at wound healing at 6 months. In the phase 1/2 study, we found greater than 90% of EB101 treated wounds reached this endpoint of 50% healing. At 6 months, about 66% of those wounds achieved 75% wound healing. The amount of wound healing certainly correlated with a reduction of pain, because always when you discuss these large wounds with the FDA is certainly used to smaller wounds from foot ulcers or decubitus ulcers that are smaller wounds, but the wound area healed in these EB patients are huge. So the wound area healed, if you actually calculate it as a surface area, it healed 130 centimeters squared at 3 months and 120 centimeters squared at 6 months. And so based on this data, Abeona was able to convince the FDA. The FDA is used to 100% wound healing. I think that makes sense when you're treating tiny, smaller wounds. But when you're treating these large chronic wounds, we finally got the FDA to understand that 50% healing is clinically meaningful. Because if you look at this data, about 53% of our patients on these wounds reported severe pain, but after EB101 application, instead of 53% of the wounds being painful, it was significantly much fewer, and that decrease in pain was durable over time. So the take-home message from this graph is we do see wound healing, we do see a reduction in pain, and so on that basis, and also on the safety data, we were allowed to start a phase 3. In terms of safety, the phase 1 was really concerned with, as a retrovirus, does this cause any systemic infection? Does this cause any immune reaction? Does this cause, more squamous cell carcinoma or anything else? And so there was no treatment-related SAEs or severe AEs. These patients, based on FDA guidance, because it's a gene therapy product, they have to be followed for 15 years. So after treatment, they're followed in person every year for up to 5 years, and then phone calls 10 years afterwards. And so what we've done is serially collect blood test for any retrovirus, and there's no retrovirus, and test for any immune reaction. And we haven't seen any systemic autoimmune reactions. There were 2 participants in the phase 1/2 study that did develop squamous cells. However, those squamous cells did not occur inside the grafted areas, and we actually took the SCC and sequenced them and made sure that there was no retrovirus in there as well. And as this audience knows, a lot of the adult RDEB patients certainly develop SCCs, and that's really related to the chronic wounding and the inflammation. So in conclusion, EB101, we think could be a treatment for large chronic wounds. And certainly in the phase 1/2A study, we saw clinically meaningful reductions in wound size, and we saw pain relief. And in some cases, the wound healing was sustained up to 6 years. There's obviously variations, and certainly for the EB101 grafts that were placed on the back where it's very difficult to immobilize, or if grafts became infected at some point, certainly the wound healing isn't as high. But I think over time, we've sort of appreciated what are the factors that optimize wound healing. And we're really hoping that the phase 3 will replicate the positive results in the phase 1/2. So what is the phase 3? After much negotiation with the FDA, this is an open-labeled interventional study. We have enrolled 5 patients so far, and there's going to be a second site opening up at University of Massachusetts. It is a one-time surgical application of these autologous gene therapy grafts. About 12 to 15 patients that are severe RDEB will be treated. At minimum, a patient needs to receive 1 wound. The maximum number of grafts is 6. So based on the phase 1/2 results, the phase 3 is focused on 2 co-primary endpoints. The first is the proportion of RDEB wound sites that reach 50% healing in treated versus untreated controls at week 24 or 6 months. And secondly, pain reduction assessed by the mean difference in the scores using a scale called Wong-Baker Facies between the treated and untreated wounds. And then other endpoints, obviously, are safety, quality of life, itch, and others. Let me tell you a little bit more about this process. So patients undergo screening. We have to make sure they meet the inclusion-exclusion criteria. We may have to make sure that they really do have enough chronic wounds. And so if that's the case, they come in at day minus 28. We do the 2 8-millimeter biopsies, send that to the manufacturing center in Cleveland, Abeona. And basically, we let the patient know when the surgery date is. And so the patient can stay in the Stanford area or fly home and come back in about 30 days. Then they are hospitalized for 7 days. They meet with anesthesia, they meet with our plastic surgeon, and basically they undergo general anesthesia where the plastic surgeon debrides the chronic wounds areas and just surgically sews on using absorbable sutures the EB101 grafts. The patients stay in our pediatric hospital for 7 days. This is because we have full nursing care to assist the patients. The first 7 days of engraftment are critical. We want to make sure that the grafts don't become infected. The pain is well controlled, and the patient minimizes any friction or scratching to the EB101 grafts. Once they complete the 7-day inpatient stay, they're welcome to go home. There's follow-up visits with a home nurse just to make sure they're doing okay. And then the requirement is that they return at 6, 12, and 24 weeks for follow-up photographs and safety monitoring. I mentioned the long-term follow-up that's required for any gene therapy trial. For the patient, there's no cost to participate in the study and no cost passed to their insurance provider. Abeona, the sponsor, will provide hotel, flights, meals, and certainly a caregiver can accompany the patient as well. And this is critically important, especially if it's a pediatric patient. What's remarkable is that, we have treated 4 patients so far since the pandemic started. And so this just speaks to how severe and serious this, RDEB is, and the patients are willing to come out. But for COVID considerations in California, the case numbers are really low, which is great, and I hope it stays that way. But no matter what, we provide a private room in the hospital, and whatever accommodations during the travel we will try to do to make sure the patient and the family are as safe as possible. And then obviously, thankfully, all the healthcare providers at the hospital have been vaccinated, and the patient will require COVID testing before the surgery. So in terms of eligibility criteria, we can take patients age 6 or greater, and they have to be medically stable and willing to travel. If a wounded area has developed an SCC, we're not allowed to treat that with EB-101, but we're allowed to treat wounds where SCC hasn't developed in those wounds. And then one key thing is there has to be a washout period. We know there are a number of EB clinical trials ongoing now, but prior to entry of VIITAL, the patients need to have washed out from another clinical trial for 3 months. In terms of eligibility criteria, a clinical diagnosis of recessive dystrophic EB, genetic testing confirming recessive dystrophic inheritance. And then we want the patient to have a little bit of collagen 7 because we don't know how patients with null mutations with no collagen 7 may react to this. And then in terms of amount of wounding, the patient must have 40 centimeters squared of chronically wounded areas on the trunk or extremities. And the goal in the phase 3 is to have matched eligible wounds. So let's pretend you have a chronic wound on the left shoulder and on the right shoulder. So that's a pair. So then we send this into our randomization scheme, and one side will be treated with EB-101 and one side will be left as an untreated control. So after 6 months at each of the evaluation time points, we can compare within the patient the amount of wound healing in EB-101 treated areas versus untreated controls. So please contact us at Stanford. If you have more questions about the trial, if you have any participants, we would love to hear from you. And so these are the emails, addresses, and the website. I want to thank all of the incredible study staff and physicians, my co-investigator, Peter Marinkovich, and Albert Chu at Stanford. We're really excited that University of Massachusetts is going to be a second site, and that'll be great for a lot of our East Coast patients as well. And all of this work wouldn't have been possible without EBMRF and EB research partnerships as well. So I think that was a little over 20 minutes, and if that's okay, I will break for Q&A. Thank you for all this time.
Thank you so much, Dr. Tang. That presentation was awesome. While I give our attendees a chance to ask some questions, I want to talk a little bit about the patient side. Do you have a sense of any reservations that are most common among patients right now as you talk to them about enrolling in the trial?
Yeah, for sure. I mean, some of our adolescent patients are in school, and especially for the pediatric patients, they really require a parent or a caregiver to be there, right? So to sign up for any clinical trial, it takes, significant time and resources, right? And so always a concern is, how many study visits will there be? And believe me, we try to minimize them as much as possible. And certainly, the critical study times are at 3 and 6 months. And the 6-week follow-up time, we have done that remotely by Zoom mainly because a couple of examples occurred where it was right in the middle of, COVID. And so
Mhm.
that worked really well. So telehealth works well in substituting some of these visits, certainly not all. A second consideration is some of our pediatric patients are quite fearful about skin biopsies, and that's
Mhm.
completely understandable. We've gotten, like others, really good at using Ativan, Versed, and other medications to really calm down the patient and to reassure them that this is a smooth process. So I have to say that overall, our patients have been pretty happy that when we surveyed the 7 patients who participated in the phase 1/2 trial, they're waiting for more grafts and they're absolutely, wanting to receive more treatment.
That was my next question, is how has the reaction been from the patients who have received treatment thus far? And it sounds like they're very willing to come back and have more treatment.
That's right. I mean, certainly, sometimes we show photos of,
Mhm.
the great responders, right? And oftentimes the wound healing was great because it was maybe at a site where the patient was able to protect the graft quite well and there was no infection. But even that picture of the upper back, that patient is desperately waiting for the phase 3 to finish. So Abeona hopefully can manufacture more grafts because so many of our patients, we're only covering, a small percentage of all the wounds on their skin.
Mhm. Are there any, have has the study included any surveys on the mental health of the patients before and after treatment?
Yeah, absolutely. There are quality of life surveys, and that data is currently being collected. So I can't really speak about some of those results, but certainly anecdotally,
Mhm.
we had one patient who said, especially on the upper back, she gave a beautiful patient webinar recently hosted by Deborah, where, just the oozing, the pain, and the time for dressing changes was so significant
Mhm.
that really in the phase 1/2, we asked the patient, where are the most significant wounds that would be impactful for your life if they were to heal? And so they gave us the worst wounds. And so we tried to treat them. And in many cases, there was great wound healing. And we had another patient who also spoke at a Deborah seminar, and he said, my feet, they're constantly wounded. I'm afraid to walk. I'm afraid to leave outside my house. And after grafting several months later, he took his first tourist walk in
Mhm.
New York Central Park. And he said, this has changed my life because now I can walk comfortably and now I can do some things that other people can do. But Jen, to your question about quality of life, I mean, I don't think anybody knows, how much of their skin you really have to replace to really make such a big impact. So I really hope that, there's a couple of really exciting clinical trials in EB, and I hope
Mhm.
they're all successful because this is certainly a very, deserving and unfortunate patient population.
Oh, absolutely. Absolutely. Okay, so a question coming from one of our attendees. Are you worried about long-term senescence of graft keratinocytes? And does the basement membrane regain normal architecture, maybe on electron microscopy?
Yeah, hi, Reed. This is a really great question. The long-term senescence is really, especially if you are putting a retrovirus in adult somatic keratinocytes, those will only proliferate after a certain amount of time and then senesce and die out. So when we are seeing wound healing this long, after a year or two, what we know is some of the epidermal stem cells must receive the retrovirus. And that's the only reason why they can continue to express collagen 7 and we can continue to see wound healing. In terms of the basement membrane architecture, it does look pretty similar to normal skin. The interfollicular epidermis looks similar. And on an electron microscopy, depending on where you sample, you certainly can see more anchoring fibrils as well. the caveat is, this is a phase 1/2 study. We only treated 7 patients, and certain biopsies and not all wounds were biopsied. So I hope that answers your question. There was another question from Zoe Smith on how do you recruit patients?
Yeah, how do you recruit patients? What are the age ranges? And have you reached out to Camp Discovery?
Zoe, those are all great questions. We recruit patients because through our relationships with the wonderful EB CRC investigators, and these are about 21 centers in the US and in Canada that participate in a consortium and a database that has identified a lot of EB patients in the US. So Sharon is part of it, Anna Bruckner is part of it and leads it. So word of mouth and emails to EB investigators, and then directly trying to reach the patients through EB Research Partnership and through Deborah. In terms of Camp Discovery, my understanding is Camp Discovery, thankfully tries to take care of a number of young patients with genetic diseases, but certainly many of the wound nurses and pediatric derm nurses in Camp Discovery take care of EB patients, and for sure we should try to advertise. That's a great suggestion. Thank you.
All right, another question. Hi, Maria. Do you think that skin biopsies are always going to be necessary? Could you potentially obtain keratinocytes from blood samples, pluripotent stem cells, for example?
Oh, yeah, Maria, I think, that would be wonderful. Right now, as a research project, Tony Oro's lab at Stanford, we are still doing biopsies, but taking fibroblasts and keratinocytes that are from adults or adolescents and putting in the Yamanaki factors to induce these adult somatic cells into pluripotent stem cells. Now you have a bank of the patient's stem cells, and you can use the same retrovirus to put in wild-type collagen 7, or potentially you could use CRISPR-Cas9 technology to correct the actual mutation. And then you would have corrected pluripotent stem cells. I think what you're suggesting is a great idea. These biopsies are so difficult. Why can't you create pluripotent stem cells from, lymphocytes? I don't know enough about that. Maybe Uni or others can comment about that. But I think those are great ideas. And I think the hardest part is, as researchers and clinicians, these are all wonderful ideas, but sometimes it requires precedence from the FDA. And I can't tell you, how hard it is sometimes to do that, especially with a rare disease.
All right, another question. For investigators interested in referring patients, what is the significance of a second site opening?
Oh, yeah, I mean, Stanford is on the West Coast, and University of Massachusetts is on the East Coast. So whatever we can do to minimize travel for the patient, hopefully that will enhance clinical trial recruitment. And Karen Weiss at UMass is a wonderful EB investigator and clinician. So anybody going to the UMass site, they certainly know EB quite well. And one thing I wanted to mention is Abeona is now trying to recruit international patients. So we're talking to a number of EB investigators from Mexico, from Canada, and elsewhere, because we certainly know of severe RDEB patients that live outside of the US. So, hopefully the IRB and everything gets approved. And, thankfully Abeona as a sponsor is very willing to, help with insurance while they're in the US, and also help with housing, because it doesn't make sense to travel back and forth internationally. And, likely the patient would stay in the US for 6 months.
All right, if we have any more questions, feel free to type them into the Q&A. Otherwise, we'll start, let's see, oh, one more from Zoe. Were special instructions given to patients after the intervention?
Yeah, for sure. So these are sutured on grafts that have been placed on really a piece of gauze. So the epithelial sheets are stapled on to gauze, and the entire gauze with the epithelial sheets are sewn on to the wounds. And so the special instructions when they go home are really, to use careful wound dressing, and not ask the patient to sit in a bath for the first 2 weeks, not have a direct shower head pressure on any of the grafts, and certainly because the grafts are placed using absorbable sutures, right, don't pull it off if it starts starting to if some of the sutures are falling out. So just kind of leave them in place. And this is why the home nursing visits and the telehealth visits are so important, because, this is new for the patient.
Okay, besides the difference in vectors, retroviral versus HSV versus lentivirus, could you briefly comment on the difference between the topical gene therapy approach of Abeona versus Crystal versus Castle Creek?
Yes, I, don't know much about some of the details, but I think, there is a theoretical concern that, retroviruses, have less control in terms of insertional mutagenesis. What we see from these 42 treated wounds and 7 patients with long-term follow-up for 6 years, we haven't seen any negative side effects from the retrovirus. In terms of the efficacy of each of these vectors, I'm not, a retrovirus or a viral expert, so I can't really comment on the efficiency of transduction. In terms of a topical versus surgical versus an injection approach, I think all of these, make sense. I do think, some of the wounds that are being treated are a bit different. And I know that for the EB101, skin grafting, it does make sense to target large chronic wounds, especially because they lead to, pain and itch, and are associated with a worse quality of life.
All right, from Emily. Hi, Emily. For the patient that had grafts on his feet, what is the time that they were not allowed to walk and the grafts were allowed to heal?
Right. From our, plastic surgery colleagues, the most critical time period for graft take is the first 2 weeks. So what we say is, 7 days in the hospital, especially the ones on the feet, please don't walk during that time. We'll help you get out of bed, use the restroom, you can sit in a wheelchair. And then when you're home, please don't use your feet and walk for day 7 to day 14. But afterwards, once the gauze has fallen off and the absorbable sutures have been absorbed, then the patient is allowed the normal activities of daily living. So, you really are, clinical trials is really a struggle. And on this webinar, know this, right? So if you heal some wounds on the feet, you can't say for a clinical trial endpoint to the patient, don't use your feet, don't walk, and don't do other things that give you joy, because that was the purpose, of the patient receiving these grafts, right? And we said to the FDA, my goodness, if, especially on young children, if we heal certain parts, they are naturally going to do more activity, and that will potentially lead to, graft friction or speed up any of the loss. But, that's okay. I mean, in the end, the phase 3 is comparing wound healing with the grafted areas versus ungrafted areas. So for that patient, if we were to treat in the phase 3 on the feet, what makes sense is to pick similar wounds on the feet, hopefully on the right versus the left. Treat one side with the graft. So the activity is similar on both feet. And let's see what is wound healing at 3 months and 6 months.
This is a great question. You mentioned a decrease in pain. Was there any decrease in overall sensation or any functional deficits?
Yeah, you raise a great point. I mean, do these grafts behave like normal skin? Are we able to feel vibration, heat, all of these things? Those are great questions. I don't know, because we didn't ask those questions. We were so focused on really pain and itch or, any, infection or anything like that. But what I do know is that these grafts are taken from keratinocytes, right? So you won't have the same adnexal structures that you would see in normal skin. So, what we always ask them is compare, pain and itch relative to, the baseline open wound. And for so many of our patients, it's the pain and itch that really is the most overwhelming sensation.
If any of you have any additional questions for Dr. Tang, you can reach us. We can reach her on your behalf through Pedra. You can email us at info@pedraresearch.org. And we can make sure we get you in touch with Dr. Tang and with the program. As I said, we will be posting this on the Pedra website, so if you want to go back and watch the presentation again, you're welcome to do that. If you haven't downloaded the Pedra app, make sure that you do that. You can do that in the App Store or Google, as this webinar and many others are broadcast live in the Pedra app, and also the recordings live on in the Pedra app. It's a great way to stay in touch with Pedra, and it's got a lot of great resources in there for all of you.
Jen, all I can say is I can't wait for the next Pedra meeting. I'm sad that it'll be virtual in 2021. I understand that, but I can't wait to hang out with you guys and have a drink very soon. So
we feel the same way, Dr. Tang. We are so fortunate to have your time discussing this with us. It's such an important work that you and everybody at Abeona is doing. We're so appreciative, and we know this is making a massive, massive difference in the lives of our patients. So thank you. We look forward to the next time we can check in on this study. And again, if anybody has any questions, just send us an email.
Thank you.